record this. Okay, thank you all for coming. Today we're going to hear from Antonin Affholder from University of Arizona in the Ecology and Evolutionary Biology group. And I don't have to read out his title, you can all see it. Yeah. So take it away, Antonin. Thank you so much for coming and visiting us this week.
Yeah, thank you, Mike. Thank you for having me and giving me this opportunity to talk about my research. So yeah, I'm at the University of Arizona in the evolutionary ecology group, departments, but I do work kind of across departments. I work also with Daniel Pai, who's at the astronomy department. We also have contacts with people in the geosciences departments. So yeah, across things. Technically, my projects right now, my duties for this postdoc that I'm doing are not exactly astrobiology, are not exactly what I'm going to present today. Separate topics that we can talk about later. This talk is going to be focused on things that I've started during my master's, worked on during my PhD, and that I keep on working on whenever I have time.
I have a lot of projects like that.
Yeah. So the title that I gave to this presentation is actually the title of my PhD dissertation. That's also maybe why it's so long. About coupling biological, interior, and atmosphere models to infer habitability and biosignatures. We'll kind of discover through this talk that I don't really like using the words habitability and biosignatures, but they are useful. So we'll have some issues with that, we'll see. Okay, so the angle I want to take to enter this is that we may already be exposed to some practical problems to solve in astrobiology, practical questions where we have some signals, some observations that we don't really know how to interpret with respect to looking for life on other planets or looking for environments that may be suitable for life. So I'll start with this example on the left, which is Enceladus, which I think is a practical case that we already have today, where we already have to make scientific decisions as to what to do with an observation, and we may have some more decisions to make in the future as more detailed and abundant observation of exoplanets might come. You know, definition of near future is plastic, so it can still be a few decades, but still that's a question that's already asked. So let's start with what was for me the beginning of working in this field, in this area, is working on the practical case of Enceladus and the Cassini mission, which I know, I'm sorry, not a lot of atmosphere to speak of, but that's going to be our entry. So Enceladus, right? It's this icy moon on the left, orbiting Saturn. Under the ice has some liquid water ocean that's erupted into space through, as these geysers here, the plumes I'm sure you're aware of. And the Cassini mission went there, took this nice picture, but also went through the plumes, the space plumes that we see, and measured some, performed some mass spectrometry on this material. In these plumes, the Cassini mission found evidence for methane and dihydrogen, which are volatiles that are pretty interesting because they're on Earth, they're tied to hydrothermal chemistry that we call serpentinization, where warm water interacts with cross minerals and releases H2 and CH4. In addition to that, other data from the Cassini mission led people who do numerical modeling of what happens to a planet as it orbits, moon as it orbits Saturn and is deformed by the tides. They predicted that there would be hydrothermal circulation, as in tidal heating, tidal dissipative heating in the core, the rocky core of Enceladus would be sufficient to power circulation of water throughout the core, throughout the porous core, and move this water around. So we have this evidence that there might be these physical processes that put warm water in contact with rock. There are this molecular evidence of potentially chemistry, hydrothermal chemistry happening. So it led people to make the hypothesis that maybe Enceladus had seafloor hydrothermal vents, just like those that we have on Earth. Not any hydrothermal vent that we have on Earth, but even a specific kind called the alkaline smokers. It's not the black ones with all the worms. Alkaline smokers, they look devoid of life, but they're actually not. On Earth, these smokers they look like this. They do harbor very rich communities of microbes. For example, this kind of microbe holds a methanogen, so archaea type microbe. And the specificity of methanogens is that they produce methane, it's in the name. These guys are even more specifically hydrogenotropic methanogens. They combine H2 and CO2 to make methane and extract energy from this reaction. They don't use light, they don't use oxidation of organic matter like we do. They use the power released by this kind of reaction to then reduce CO2 into organic matter, perform functions, mobility, whatever. And the same paper that was analyzing CINI data for these volatiles, they tried to infer or calculate the value of the Gibbs free energy of this reaction in Enceladus' ocean. They predicted that this the affinity or the negative Gibbs free energy was positive, meaning that the reaction could yield some energy. So what do we conclude with that? What practical decision do we make from that? We see energy, we see resource for some microbes that we know on Earth. Do we conclude that this environment, and so this ocean is a good environment for them to grow in? Or do we ask, would we see the resource if there were any of those organisms? If there were some methanogens, wouldn't they consume, H2? And in consequence, we wouldn't see it in the plume. Would they spend the chemical potential, the disequilibrium, until it reaches closer to equilibrium? In which case, we wouldn't be able to see Gibbs free energy available in the ocean. So we need to do something here. We need to kind of crunch numbers and know what happens, what's the minimal energy they can survive on, and what's the leftover energy that they would leave, what's the leftover H2 they would leave, what amounts of methane can they make. So that's where we thought, Regis, that we could do something as theoretical ecologists, where there is a strong culture of resource population interaction, modeling this kind of processes. So we try to turn this problem into, a mathematical language, right? The goal that we want is down there, maybe not so conveniently, but we want to be able to predict the value of some observable under some hypothesis. In this case, the observable is the amount of methane or amount of H2 in the plume of Enceladus. And the different hypotheses, I'll get into that. So first, what we do when we're a theoretical ecologist, we write a derivative for a population size. Usually, the models that we use, we try to find out a way to express a per capita growth rate. That's the G function there. That is then multiplied with n, the population size. This per capita growth rate is function of some environmental state that I know E here that can contain many things, temperature, abundance of resource, things that will change, things that will not change, and then potentially n, the population size as well. One thing that we can find in the theoretical ecology literature is something called the invasion fitness. It is this expression here. So the per capita growth rate when the environment, the local environment, it's at a steady state that is a zero population steady state. So take the abiotic steady state, no biological process whatsoever, just take the abiotic processes. What's the state of the environment? Is the per capita growth rate positive in this condition? In other words, that means that the zero population steady state is unstable in the dynamical system. Or in other words, again, that's the theoretical equivalent of putting a microbe somewhere and it would turn into a population. It would explode into a population. So that's invasion fitness, invasion criterion, viability criterion, whatever its name. We are going to use as the unit of measuring what's a good environment for the microbes. We'll figure out the G function. And then what we want to do is to solve this local feedback between the population and its environment, or rather the resource abundance or product abundance, waste abundance rather. Solve this the equilibrium between those two to predict the state of the environment at equilibrium and then try to also add some component to translate environmental conditions or resource abundance locally in the hydrothermal vents with the observable that is in the fluid and space.
Can I have a question?
Mhm.
So this equation there doesn't have any sink term, so it's exponential growth, right?
Well, provided, I mean, the G function can reach zero at a certain.
So it is
even-
like a complex function that
Mm.
has a time dependence and it, or even,
Doesn't need to
So-
have a time dependence, but there can be, what we call density dependence through resource depletion.
okay.
If the population
Yeah.
consumes resource, its growth rate might go, per
Mhm.
capita growth rate might go to zero.
Mhm.
Can also have death rates. Yeah, it can be a function that has some fixed
Sinks,
sinks. Yeah,
some sinks of
Mhm.
that, okay.
not necessarily,
Okay.
it doesn't have to, but it
Mhm.
usually does, yeah.
Yeah, otherwise I don't see how we can get equilibrium from that.
If it goes to zero at some point. Okay. Back to our practical case, and for us. So, or maybe I should rather start with just a little bit of ingredients list for our recipe. We still need a bunch of components. We need to figure out the G function, what's the growth rate of these organisms. We need to figure out, a way to constrain this term, what's the abiotic state of the system. That leads us to a nice problem, and we need to figure out the relation between the local environment and the observable. So what was in the flume. Interestingly, this term here, satisfies this term here, bio, the biological equilibrium, when the population is at equilibrium with the environment, this system state satisfies the G function being equal to zero. So it is a limit of this criterion. What it means is that an environment at equilibrium with an organism will have just barely enough resource for each individual microbe to grow. So, back to our practical problem from earlier, an environment that has a population doesn't look that good for a population. Think about a pasture with cows, without cows, right? Without cows, you'll have, tall grass.
Yeah.
With cows, very short grass. So that's kind of stuff. Okay. That means something as well. That means that we really need to a priori build the local abiotic environment to assess, the viability of the population. If we use direct inference from the observation, we risk, if there is lag there, there is a conflict in the way information flows, right? I don't know if that's clear, but it goes around its tail, is what I mean. If we directly try to infer the value of the environment and then conclude whether it is habitable or not. So yeah, back to the ingredients that we need. So we need growth function. So here I note B because I count biomass, not cells. So that's the usual, notation. What we used is a model that's focused on the energy that's available. So still continuing iterating on this question about the Gibbs free energy of the reaction. We just have a way to constrain a little bit better what's the minimally required energy for the cell to grow and how this minimally required energy changes with, for example, temperature. Well, in this case, only temperature. And we can tune the parameters of this model to fit the growth profile of an existing organism, with a maximum temperature for growth, a minimum temperature for growth, and an optimal temperature for growth. We also wrote a very abstract, very simplified model for mixing of hydrothermal fluid around the vents that composes the local environment. So the composition of the local environment will be function of composition of the ocean on the one hand and composition of the hydrothermal fluid on the other hand. And that's when we have our last component, which is the component, the geochemical constraint components, where these compositions are constrained by some geochemical models. Models that were focused on the pH of these oceans or knowledge that we have on Earth of what can happen when serpentinization occurs. How much H2 can be produced, so up to how much H2, up to how much CH4 is still. And then last, conveniently for Enceladus, kind of a standard model is that the mixing of hydrothermal fluid at the bottom of the ocean gives, this warmer water, buoyant water that rises and that stays in a neat column all the way until it reaches the ice. And thermal models, they predict that the hydrothermal circulation would occur right below the big plumes that we see. So what happens in the hydrothermal vents is assumed here that to directly feed the plumes that we see. Then you have a lot of problems with exactly the composition of the plume compared to the composition of the water, because many things can happen, when the ocean is outgassed, etc. So what we focus on instead is the rates of escape of the hydrogen and CH4. So assuming that the ocean is kind of at the steady state, that whatever escapes in the plume is compensated by something that's produced inside. That's the assumption to build this observable.
Are these, you said that these holes where the gas jet is, are exactly above the vents? Is this the thermal circulation perhaps that melts the ice?
Yes, that's very much the
Yeah.
standard model for.
Okay, can I ask a question too? But you said you assume maybe steady state, but Cassini made many flybys. Were they able to put any constraints on if there was a change in the flux for these molecules? No.
They don't seem to change by a lot, but also the flybys were not super far apart.
Okay.
So it is not an uncommon hypothesis, to say that there is a steady state, but there's not compelling evidence to support it.
And what are these terms in this equation?
So these terms, okay, so here we have the energetic yield of the catabolic reaction, so turning CO2 and H2 into methane. Here we have the costs of building biomolecules. So, how many kilojoules of energy do I need to build 1 mole of molecules in the cell on average? So that's a term that's called usually the growth yields. How many times does the reaction need to run in order for the cell to build 1 mole of biomass? Then you have this term here, which is a maintenance cost. It's spending of energy for the cell to maintain itself. Usually it's, assumed to increase exponentially with temperature. The higher the temperature, the faster all of these destructive processes that happen in the cell occur, and the cell, dies because it's not able to replace those molecules anymore. And then there is this term here, which is so usual. It's kind of a fine-tuning term. It's a fixed death rate that represents maybe, deleterious mutations or external events that lead to cell death. And that's usually required for the model to behave nicely.
Yeah, that's why you get a steady state.
But we don't need it. We still have a steady state because this is directly coupled to environmental. It's directly a function of the concentrations of H2, CH4, and H2, CO2, CH4. And as those get depleted, this might take a special value where this and this are equal. So you still have a sink. But this one is regarded. This one is actually pretty low. But it's the thing is, if we don't have this one, the minimal temperature does not exist. Because those two are really exponential function. Well, it's technical. But if we want to put a hard minimal temperature bound, we need this G term. Okay, so let's see what we simulate. So those are simulation outputs. We simulate over ranges of H2 production by hydrothermal circulation, CH4 production, a lot of different parameters that we scan over. And we get these distributions. So this is flux of H2 and this is flux of CH4. The magenta lines and stars are the observations made by the Cassini mission. And we sort our simulations into 3 cases, right? We have the blue and orange cases that are abiotic, we just don't model a population there. The specificity of the orange simulations is that our model predicts that a population could grow in the conditions that are simulated. We don't simulate the population, we just measure that it could grow. Habitable, but uninhabited. So what we see from these results is, first, we're able to reproduce the observations, always nice. but under only one subset of simulations, the biotic subset. And that's solely because of methane. Our abiotic model is unable to produce enough methane to match the observations. In our abiotic model, we just have as much CH4 as was observed being produced abiotically in serpentinization experiments. That's the only source of methane that we have in our abiotic model. So conclusion, this abiotic source, not a good explanation to data. On the other hand, simulations with biotic methane productions, they do reproduce observations. So, you know, based on that, on the methane flux, we can't really exclude the possibility of biotic methanogenesis studies. But then what about the leftover resource that I was mentioning first? If we look at the green and the orange distributions here, they're not too different. So it turns out that the population of methanogens does not consume that much H2 in the end. Not enough to make a significant difference that we would notice from observation. So in a way, the abundant H2 that we observe is still compatible with, conditions that are favorable for methanogens growth, but also compatible with consumption of methanogens.
Can you please explain the graphs one more time? So when I see the distributions there on, let's say, panel A, this is the flux of hydrogen, right?
Yeah.
Uh,
So A is flux of hydrogen distribution.
yes.
B is flux of methane, and this is just a combined.
Okay, so when you say that the planet is, or the heavenly body is uninhabitable, then the simulations show that you would expect a hydrothermal flux of order 5.
Yeah,
moles
those
per
are
year?
locked in. Mhm.
Okay, but when it is abiotic and habitable and biotic, it would be 10 moles per year. Oh, no, 10 to the power 10 moles per year.
10
This
to the
is-
10, yeah. Well, up to 10 to the 10. So kind of consistently with what we might expect, if we were to observe, low H2 production, that's not incompatible with like weak serpentinization happening, but it's not enough according to our model to fuel methanogenesis. So that would be the uninhabitable blue distribution.
So this hydrogen would be the leftover hydrogen with a biotic life, or would this be the minimum hydrogen that you would require from the hydrothermal vent to be expelled to sustain a biotic signature?
So that would be the orange distribution. That's kind of the same as the green distribution. We don't see it that well.
Okay, so this graph tells me that this is the minimum amount of hydrogen that I need.
So that would be, this, if we were to observe anything less than this amount of methane, of H2.
Yeah.
Assuming methanogenesis would be a bit harder to observe,
Okay,
I think.
but the biotic flux of methane is close to order 8 to the power 10 moles per year. And your hydrogen flux is also, the peak is close to between 8 and 10 moles per year. But the initial reaction that you showed me, the hydrogen and methane should be in the ratio of 4:1.
Yeah, that is
For methanogenesis to sustain. So if I'm seeing as much methane as hydrogen, what does that mean? Where's the rest of the hydrogen going?
it's just not being used, maybe. But also, I mean, okay, let's also acknowledge that there's not much detail on my scale. So a factor of 4, I don't know how much I would see it.
No, it's a linear scale.
Those
It's a
are
logarithmic scale.
logarithmic scale. Yeah, so a factor of 4, would be half an order of magnitude, and have a 5 order of magnitude on this distance. So yeah, I don't know exactly what's the delta, what would be the delta, but of course, initially people would try to see if we would see such a ratio between hydrogen and methane. But if a lot of hydrogen is produced, but not a lot is used, how much a population will impact the ratio hydrogen to methane is not super clear. I have an additional figure where I show the ratio under the different cases. I don't know it by heart, so I can't really make the analysis right now, but yeah.
No worries.
Mhm.
And one question about the H2 supply. So in hydrothermal vents on Earth, do we see that fluctuating much in vents, or is it within a factor of 2 of the mean, or what?
So that's not super well known because
It's not super well known.
so where we can constrain the concentration of or the production of H2 is when we do lab experiments of self-optimization.
Okay.
So those, are controlled and
Yeah.
flux is going to be pretty stable. If we go to the ocean and people do that and grab samples or look in situ, I'm not exactly sure. There is definitely a lot of variability in the temperature of the fluid.
Okay.
Yeah, so completely messy, right? So our idealist view of equilibrium or neat little gradient around the hydrothermal vent is it's a complete made up story, right? Now the question is how bad it is that it's made up like this.
It's not a made up story. So we measure the amount of water which is injected out of a hydrothermal vent, assuming that it's going in. And I'm only talking about the axial flux, which is shooting out of the vent like an injection. And then we take samples around that and measure the concentration. So with the product of the concentration and the flux coming out, you measure the expected moles per year, and that varies between 0.3 to 1.5 times 10 to the power 10 moles per year. So it's a variation of 1 order of magnitude in hydrogen.
That's a lot.
And this story applies to all elements, iron, methane.
Oh, they're all varying by order of
Yes.
magnitude.
And this is only along the axial flux. The
Right.
off-axial
You
flux
don't,
are even
No
more.
one's measured that.
Yeah,
Okay, I see.
Yeah, so now
Jeez.
a question is how much this kind of variability would affect this kind of volume. There's not a lot of idealized modeling of hydrothermal vent environments to put populations in there. There's been some work, to look at convection, transport, diffusion, whatever models in the chimney wall. Not as much the broader environment. So that's it's a big deal. Yeah, let's ignore kind of the takeaways there, but maybe just to focus on the pending questions that I can think of right now is. Sure, okay, H2 produced by self-optimization, but the core of Enceladus is pretty small. And the ocean circulates through it pretty fast, all things considered. And Enceladus could be youngish, could be older, we don't really know. And there's been, studies that suggested that self-optimization wouldn't be sustained over super long periods of time. So, is that the end of the self-optimization hypothesis? Do we need to come up with something else to explain H2, in which case this whole scenario goes down? And yeah, well, I think, sustainability questions, closed environment like this, like a small icy moon that has an ocean that's covered with ice. And that also connects with maybe carbon cycle questions. The implications of assuming that there might be a bias here, what happens to carbon over long periods of time? Because there's no plate tectonics where you have sediments that would be exposed to air again or whatever. So it raises, I think, interesting questions on the sustainability of biosphere in this kind of environments. Okay, let's move on. So great stuff. Icy moons. In my introduction, I had kind of two practical cases. One that's very real, that's Enceladus that we have measures of, but also the second one would be terrestrial exoplanets in the future. And now I want to use some methodological elements that we had explored for the Enceladus case and go on to exoplanets, terrestrial exoplanets to see what we can do with those methodological elements. So yeah, this blah blah blah. There might be better telescopes in the future. We might get some atmospheric spectrum for sighting, but also what do we do with them? And first with Enceladus, I was in my the methodology that we developed for Enceladus, we had to build some sort of abiotic model first, right? To evaluate the biotic model. So I tried to do this for kind of Earth-like planets. I mean, planets, one Earth mass planets orbiting Sun-like stars. I'm trying to kind of look at different, at least insolation values, but in our mind, those are, Earth-like exoplanets that we tried. And one problem that we that we tackled to enter into this world was, it's kind of textbook knowledge that the carbonate silicate weathering cycle is required for Earth's habitability over the long term. But other terrestrial planets in the solar system, they won't really have this happening, this geological carbon cycle happening right now. They might have different things happening way back in the past, but probably for a long time. So it kind of raises the question, all or most planets like the Earth with a plate tectonics and carbon cycle, or are they, mostly like the other planets of the solar system without this thing? And the way it matters is that one, extreme way of thinking about it is that a planet that doesn't have this plate tectonics regime, the alternate regime that we call mobile lid, they wouldn't have continental crusts, so they don't have exposed silicate to the surface as the Earth does. There's no resurfacing of sediments, to resupply minerals, but also carbon. There's no recycling of crust-bound carbon through the mantle to fuel further outgassing later down in geological history. And these processes, they matter for the equilibrium concentration of CO2 in the atmosphere that's regulated on the one hand by outgassing by volcanoes, and on the other hand, the precipitation to carbonates, basically, or solubility in the ocean. That's on the very long time scales. That's on tens of millions of years time scales. So yeah, we have an image of kind of two unmemory scenarios where one planet has continental weathering, the other one does not, when it's in the stagnant lid regime. I think I mistook stagnant lid and mobile lid earlier. Mobile lid. Plate tectonics is mobile lids. No plate tectonics would be stagnant lid. I will confuse them more, I'm sure. So that's for the weathering parts. We still assume in our view that there is seafloor weathering happening on the stagnant lid planets, because weathering of basalts can still participate to some extent to the carbon cycle. And we also assume different scenarios of volcanic outgassing where the mobile lid planet would have sustained over a long period of time volcanic outgassing, whereas stagnant lid planets would have only a lot of volcanic outgassing early on in their geological history. And now on these different scenarios, planetary scenarios, we want to basically integrate that system. We have a set of variables, a bunch of molecules in the atmosphere, H2, CO2, CH4, molecules that are like, we also have CO. And then we have these variables here that are ocean concentrations. So C is total carbon, total dissolved carbon, DIC would be also another name for it. Alkalinity, it's a complicated concept, but basically it measures excess positive charges that is delivered to the ocean. And so we have two layers in our model ocean. We have an upper layer noted by O and a deep layer noted by D. And they mix together. But that's what we do. And when I talk about the partial pressures of these gases, I'm really talking about at the very bottom of the atmosphere, the first layer. Yes.
Why do you say that the C value there is DIC? You exclude the option?
No. DIC, dissolved
inorganic carbon. Exactly. So why don't you exclude the dissolved organic carbon if you're looking for light?
Okay, no, right now we're building the abiotic scenario.
Okay.
So we don't put light here.
Okay.
Next slide. Well, if only. Also, yeah, I'm sorry, there's a lot of equations maybe, but so we do model, ocean atmosphere exchange, and there can still be a flux and out of equilibrium situation between the ocean and the atmosphere in our model, provided that compensated by other, sinks and sources. Okay, we put some, continental erosion plugs, weathering plugs, seafloor weathering. There's separatization is involved for H2 that I don't show here, but yeah, basically we have these differential equations that involve these big classes of processes: weathering, outgassing, exchange with the atmosphere, precipitation of carbonates, and mixing of the upper and deep ocean. This model was developed by Joshua Christensen Totten a few years ago with David Kathleen. It's one model, one parameterized model looking at the carbonate cycle. I'm sure there are, others.
There's no advection also, right? There's no horizontal advection.
No. It's a 1D model with two boxes, maybe, a point of a detail that's maybe useful is that here you see that it's just aqueous CO2 that's going to exchange with the atmosphere, of course. So we do solve the carbonate chemistry in the ocean. Just assume that it's always at equilibrium with respect to the GIC, alkalinity, and atmospheric CO2. Yes. So that's for the kind of ocean carbon cycle component. Now, the atmospheric components. I'm nervous now to talk about this. What we do is that we use 1D part-3 model that's called Atmos, developed in the casting Pope. We use the climate part to get a temperature from forced composition of the atmosphere, so forced bottom layer composition of the atmosphere. And also by forcing this composition, we look at what are the projected photochemical rates that are calculated by the model when it's at radiative equilibrium. And we run this model over a grid of different conditions. By different conditions, I mean different values of these partial pressures. We also vary the luminosity S. And then we interpolate over that grid. That's a 4D grid. So interpolation, is not perfect, pretty hard actually, but we use it. On the right-hand side here, I have just a showcase illustration of the calculated temperature for different PCO2s, PCH4 at a fixed Archean-ish S value. So that's the luminosity fixed pH2. And you notice that there is a huge gap here, which is probably the haze condition, I guess, when there's a lot of methane compared to CO2. So we don't do hazes. We don't go there. We don't want to look at hazes. So if the model for some reason tries to go there, we just stop the simulation, label it, maybe we'll go to haze and move on. On the left-hand side here, I have photochemical fluxes in different conditions. Maybe one thing that's interesting to note and that maybe we'll discuss is we do have a non-zero CO2 photochemical flux in the model. In undergrad, I always thought that, CO2 and photochemistry, nothing to do. But this model predicts that it's a very low flux compared to the rest, very low. But it ends up mattering, as we'll see, when there is nothing else happening to CO2. On a planet that has no volcanic outgassing, no weathering, in the model, we only have photochemistry that would affect it. So even a low, tiny amount of photochemical rate for CO2 will end up structuring the data that we see, which is basically this weird thing that we see here. But okay, let's skip that for maybe discussion if you want to. So left column is simulations under the hypothesis that we have continental weathering, that we have long-term outgassing. On the right-hand side, it's the stagnated planets that doesn't have this continental weathering. The color of the dots is the age of the planet, where we basically have a parametric relationship between the age and volcanic outgassing. So that's the only thing that matters. X-axis is basically relative luminosity, and y-axis is the PCO2. On the left-hand side, we see a very clear, decreasing trend for PCO2 with luminosity, which is what one would expect, from the carbon and silicate weathering cycle. But on the right-hand side column, it's more complicated because we do have one set of simulations that has this kind of relationship. That's the blue dots mean young planets. So again, we kind of expect it because young planets, they do have volcanic outgassing, they do produce basalts that's going to be available for weathering. So kind of behave as expected. What's not maybe as much expected is all of these dots, which is this quirk that I was mentioning, and the spread that's there. Yeah. Some conclusions that or interpretations that we draw from this is, I should also say that under our model, most of these simulations are actually separate, even in the standard case. So interestingly, if you make the thought experiment that you sample terrestrial exoplanets in the habitable zone of sun-like stars with uniform age, whether whichever interior convection regime is most frequent might not matter as much for the frequency of these planets that we expect to have temperate planets. However, it still matters for, considering the long-term habitability and the billion-year scale evolution of these planets. So we still ask, given that we have two somewhat different distributions under the two scenarios, how can we, know which distribution is true when we observe planets? How many planets do we need to observe? What's a resolution that we need to observe them at to infer whichever scenario is true? By scenario, I really mean a view of the mind that either all planets are in the stagnant states or all planets are in the mobile state. And we measure as a function of the minimum CO2 that can be detected in their atmosphere and the sample size, we measure the probability that a sample would yield evidence for the wrong hypothesis. And we know what is a wrong and a true hypothesis because we make up the data ourselves, right? So we simulate and then we just make a fake statistical test and evaluate the risk of these tests. And what we see is that for 25 exoplanets at which we detect, and if we're able to detect CO2 if it's greater than 1%, then we actually might, in 95% of the cases, have good evidence in favor of the true hypothesis. So, the HWO target yield is not maybe, it could be sufficient, provided that we are able to measure 1% CO2, and that the 25 exoplanets are actually, Earth mass planets around a sun-like star, which they're not going to be, but we don't know how to quantify, how things are going to, vary with this because we don't have this generality in the model. Okay, quickly move on to life.
One
Yes.
question about the Atmos application in the last section. Do you use a different geological setting of Atmos over time?
No, we
Which-
fix
Which setting are you using? The Earth, the present day Earth-like
No.
setting?
Arcane Earth.
Archean Earth setting. But if
Yeah, Arcane Earth. So we do have, for example, we vary the continental fraction, but we don't vary it for Atmos. We vary it, for the amount of weathering that's going to happen in the kind of the geo box of the model. But yeah, Atmos, as long as Atmos is concerned, they're all Arcane Earth planets.
there's more of a comment than a question in that case, because the species themselves would change if you were considering a different geological age. The species in the atmosphere, not the atmospheric composition.
Yeah.
Over Earth's geological history doesn't remain the same. So if you did these simulations, keeping everything else same and plugged in a different era in Atmos, you would get different
Yeah, most definitely.
concentration.
So yeah, I think, the base template for this model was the Arcane Earth.
Okay.
So even though we talk about billions of years, it's not entirely, unintentional either. Because, from our biological perspective, the big innovation, of course, is oxygen at some point that goes into the atmosphere.
Can you repeat that, sir?
Well, for our perspective of biologists, one big dramatic event that happens in the atmosphere, of course, is the great oxidation with, oxygen being there. And we just kind of asked the question, what would happen to the planet where, we don't have this because we're really in an abiotic case. And if we put some oxygen, we really want it to be something that's going to be a biotic process. Okay. So actually, I'm going back in time because what I presented earlier was this whole last section was some work that we just submitted, so it's also pretty still not as mature as maybe the rest. But now we're going to move on to life, including life in those kind of models. And we did so for the specific case of the Arcane Earth. It matters, there is this trade-off between what we can do with the biology and how generic the model is. This atmospheric, we are already using a super simplified version of atmospheric processes, and that's already difficult to do in such a general way. It's much easier to take just the Arcane Earth, just a few 100 million years, fix a lot of things. That's already much easier. So that's what we started with, and we simulated early kind of biospheres on this Archean Earth model, where we do have a carbon cycle model. We do have the parameterized climate model. We actually use this time outputs from the NMD GCM that we interpret. And then, just like the same architecture of models that I showed you for enceladus, we use for methanogens, so the same kind of microbes, but also other microbes that perform different reactions. The common point between all of those is that they don't use light, they don't use oxygen. Okay, those are early microbes, chemoautotrophs. We run simulations there, and one thing maybe that's interesting to us is when we look at the equilibrium atmospheric composition under different scenarios of metabolic assemblage. So these are different colors. For example, yellow would be, or yellow-orange would be only methanogens. Blue would be abiotic. Green would be those cetogens here, Ag. So distinct atmospheric composition for different metabolic assemblage scenarios, right? So that's, I think, an interesting thing to explore when we think about biosignatures and inverting the problem from atmospheric composition to whatever happens, on the surface from a biological perspective. Maybe, a different kind of interesting that we observed in this model is what happens to climate when new metabolisms are included. When basically methane producing organisms are included in the model, suddenly the temperature rises because methane is produced. Then there is this compensation time, which is the carbonate silicate weathering cycle. And if and when methanotrophy evolves, methanotrophy consumes methane, then methane is removed from the atmosphere because of that, and the temperature drops. So it was nice to see happening this kind of destabilization from within, from the biosphere that could warm the planet or cool it, depending on what happens, what evolution makes. It's that.
What's the time scale for between?
Sorry, it's part of a larger panel, so the x-axis got lost. I guess it should be a few tens of millions of years, if not 100 million years. But yeah. Off the top of my head, I would say at least 50 million years. On the right-hand side of this panel, where we measure probability of glaciation as a function of the time delay, because the time delay between methanogens arising and methanotrophs being evolved kind of matters as well. I'm not going to detail that too much. Okay. Another scenario, another case, Noachian Mars. So that was more recent, that came out 2022, late 2022, where we used the kind of modeling that we do for microbes for Noachian Mars. Except this time, we don't really model the biosphere in the ocean. We model it in the porous crusts, in pores that are filled with brine in the crust of Mars in the early times. Since I'm here, I'm always giving those details about the kind of atmospheric models that we use. Here, it's even a different one. It's a parameterized version of the DPL 1D model. So it is a 1D model. Even the biological model, of course, is 1D in the crust. It depends. The results, they've been expressed as a function of different values for some relevant parameters, such as the freezing point of brines on Mars turns out to be of great importance. I'm only showing results for a specific case, but of course, just a specific case where the brines freeze at 252 kelvins. And so interestingly, we calculate that the possibility for the methanogens to grow in the crust is kind of limited by the ice cover. If it's too cold, you're gonna have ice that insulates the crust from the atmosphere. And thus, even if there is H2 in the atmosphere, it's not going to be available for methanogens that are under the crust. And curiously, perhaps, those methanogens on Mars, by depleting H2, that's kind of hypothesized as a way to have temperate early Mars conditions. So the depletion of this H2 would not be sufficiently compensated by production of methane and would lead to cooling of Mars. So the same metabolism that was leading to temperature increase on Earth, on the Archean Earth, same exact metabolism, we calculate would yield to, well, of course I'm getting mixed up, but would yield to cooling, to case versus the initial condition, lead to cooling of Mars. What I originally wanted to go with on this slide is that we also still see that there is somewhat of a difference in the atmospheric composition in this model when we have methanogens and when we don't, mostly for methane. And that's despite the fact that we calculate a very, very small productivity of the biosphere. By productivity, I mean the amount of biomolecules that are produced.
Doesn't that mean the time scale is quite long then? Could
So,
be
yeah.
from the initial delivery of the H2 from impactors until the methanogenesis takes off and starts consuming the H2 and then drops your temperature. So that lag could be long if the productivity is low, is that right? Or do you think it's?
That's a good question that I don't think we have the answer to. Or maybe I don't, maybe Bowie studied author has it, but
Cool.
good question, yeah.
Just because you had sort of a timescale in the previous process.
Yeah,
I just thought you might have thought about
I
that.
think the way we do it here is different. We don't have an explicit timescale in the model.
Okay.
We just go to solve the equilibrium. So yeah, what we did is produce nice visuals by projecting a 1D model over whole surface. No, but because basically in the model the depth, we calculate the depth at which we might find methanogens because matters with the pressure and the temperature profile that's inside the crust. And so one of the discussion points of the paper was to discuss what could be a good place to look for if this is true, if Newark and Mars could have methanogens in the crust. So where would you have to drill the least amount on Mars to find them? So yeah.
So does that mean you're looking at areas that you think had some surface water for some periods of time? Like, I mean, Perseverance is in Gale Crater, right? Is that a good place?
It is a good place. Yeah.
Okay. Just,
It's in Jezero.
oh, sorry. Yeah, sorry. the other
Perseverance
ones.
is in Jezero.
Yeah. So
Yeah.
curiosity's in.
So
That's right.
would someone know what this is? Is this Jezero?
That's Hellas.
That's Hellas.
Hellas.
Yeah.
Jezero is right above that.
And it's a really, really small component.
That's Jezero.
Okay.
Or Jezero's on the like the northwest of that.
Tiny.
Yes.
Okay, it's tiny.
You can't see it.
Okay. Yeah, Boyens has another figure that I've included here where, we have the map of the most optimistic depth at which we find microbes in our model and sites at which, probes have been sent, where the robots are and where future missions will be headed
So
out.
optimistic depth, you mean not very deep. That's what you're saying.
Yeah,
Okay.
yeah, at best, but it's still like a kilometer.
A kilometer. A kilometer. That's optimistic.
It's
Okay,
of the
wow.
order of a kilometer. So in the end, it's not that optimistic. So yeah,
Send a big drilling platform.
yeah, the drilling set of things.
Sometimes we get there.
Yes.
Uh,
We need Starship.
So why such deep depths are more favorable? Because it's warmer there? or-
okay, I need to open the article, I think.
Well, we can discuss it after on. once you
Okay,
finish.
let's do that. Yeah, I will need to pull up the paper again. Yeah, so okay, takeaways from this part. Yeah, in our model, it's pretty frequent to find depths, even if it's great depths sometimes, at which the pores still exist. They're not crushed, by hydrostatic pressure. They are filled with brine that's not frozen, and they have access to enough H2 to grow methanogens. That's still not a rare occurrence in our model, which is perhaps surprising. Methanogens significantly change the atmospheric composition under our model. So again, the biosignature connection, and they have a kind of a negative effect on the climate. So that's also interesting for, what's the role of an early biosphere in the climate stability and such of early planets maybe. Kind of question. And I think, yes, that was my last slide, where I'm going to leave you with a summary that you will read or not, but mostly I will discuss the future directions that I want to take from there. So of course we have elements of a generic-ish model for Earth-like exoplanets. We have elements for modeling the biosphere and coupling it to the kind of models obvious next step is to, try to apply that for exoplanets and see if we can see patterns of atmospheric composition that we can also try to tackle as population scale, hypothesis. I mean by that, what I presented for the interior convection regime, I make population scale hypothesis, all planets are in that regime, or all planets are in that other regime. And that helps us, trading sample size with resolution of the observation to get something out of it. We want to do that with biosignatures as well. Can there be some trends that we can predict, from coupling these models together, and that we can use to inform what could be done with future missions. Then one thing that I really want to do is what I was mentioning last is kind of quantify from a dynamical perspective the resilience of the climate city states. When you have life, when you don't have life, when you have this kind of metabolism, this assemblage. In a certain context, Mars and Earth, we saw, different contexts into different outcomes with the same metabolism. It's been decades since people have hypothesized all sorts of Gaia kind of hypotheses. Of course, now we have at least tools to run more complex models that are still idealized, but more complex models, and see if there is anything as a generic trend, a structural trend for these models that include life or not. My guess is on not, but we have to do it to know it. And yeah, thank you so much.
So they have to go, I have another meeting, but I'll see you tomorrow.
See you tomorrow.
Okay. Any other questions in the room? In addition to what's already been asked?
Yeah.
You have,
Go ahead, Igor.
I was just wondering, like, you have, you describing hydrogen consumption on Mars by this, but hydrogen also escapes pretty quickly. So, did you estimate what the competition between hydrogen escape and consumption by the biomass? Maybe it will escape quicker than your biomass is consuming in that sense.
So of course, over the long time scale, it's going to matter because at some point there is just no more hydrogen in the system. So we do have hydrogen escape in the model, but we look like the short time scale equilibrium where, basically outgassing of H2 is compensated by both escape and consumption.
Yeah. Okay, so you assume that basically that you have equilibrium. Because I guess about hydrogen, some people say that it's impactors, not outgassing.
Right.
So it's kind of never in equilibrium in a sense, I guess.
It's in the epoch. I think there's more volcanism early on, where probably H2 comes out in the earlier part of Martian history, but by the experiment, I think volcanic outgassing is lower. Although, regardless, it appears you need impactors to help with the climate. Right? But in the time scale of equilibrium, people have to calculate that.
Yeah, but
Just
anyway,
making sure,
I understand
the
that
underlying
you
hypothesis
basically
is volcanic outgassing.
you just assuming equilibrium. But just in your equilibrium, I'm just wondering, do you remember numbers, like how much is being lost, how much is being consumed? Like order of magnitude, is it the same amount, order of magnitude bigger, smaller?
I don't remember.
Well, if you don't remember, that's fine.
We should discuss it later.
Yeah, I'm not even sure we discussed that. Honestly, Question.
Call show.
if others are,
Oh, so what did you have?
you used a coupled atmosphere ocean climate model, right? And I saw that you considered photochemistry in the atmosphere. Did you also consider photochemistry in the ocean then?
Actually, yes. Yes. I'll go back. But if you look at this slide, I said we had PCO in our model, but I did not discuss it. Carbon monoxide. We do have a module for photochemistry of carbon monoxide in the upper layer of the ocean, surface ocean.
Okay. Does the change in mixed layer depth change your results in that case?
I don't know.
I don't, they're not using a fully coupled ocean.
It's a 1D model, so there would be a mixed layer.
There is,
It depends on what they're,
Definition of 1D is, yeah.
yeah, we're using, do you know this paper by Current Chat, 2005? Where they do a coupled model with Atmos. They use the mixed boundary layer model where it's not, you know, in the modern ocean, I don't know, the mixed layer is maybe 50 meters, 100 meters, yeah. I don't think that's what is done in this model.
Okay.
So the idea is basically you have a tiny film
Okay,
that's at equilibrium and then, you know, diffusion.
thank you.
let me go back to your Mars question. Do you have a question?
No.
So with your Mars stuff, you were talking about Milwaukee and these, basically very small amounts of water. Basically, I think you're talking about sort of icy highlands hypothesis that goes to a short period of warming. So you get some, I guess, lakes and stuff like that. So that's where the brines and the water are mixing and coming from for your model input, I guess. But, you know, there's another hypothesis that you could have this possibly short-lived northern ocean. So if you think about that scenario for a minute, what do you think the observables could be there? So maybe you don't have to go a kilometer down if the ocean was living long enough, and therefore you don't have as much settling, or I don't know. What do you think? If that hypothesis is real and we eventually we have, I mean, there's a Chinese rover there now, but it's not digging like Perseverance or Curiosity is. So what do you think about that?
Yeah, good question.
Okay, you don't have to
So
answer it.
Enthenogens, they don't leave a lot of, you know, they don't leave a lot of trace. So these kind of microbes, Archaea, Enthenogens, very tiny cells. I'm not even sure if we are sure that they fossilize. Because we have found stuff that, you know, look like fossils of this kind of cells, but then people have said, no, you can produce them abiotically. These are fake fossils. So there are even debates there, and they don't, you know, they're not algae, they're going to precipitate carbonates, and, you know, enhance carbon precipitation. So what kind of evidence they would leave behind is not, you know, it's a research question.
But you can't look at carbon isotopes to figure out what
Yeah,
are
I
these
think,
things?
I think
Yeah,
you
it
would
is.
wanna do isotope.
Yeah, but in what thing?
I mean,
In the
any
substrate.
organic carbon that's there in the substrate.
Yeah, so when you did your core, you would look at the
Yeah.
isotopes that come out of the core.
I mean, they haven't found a ton of organic carbon from Perseverance.
Okay,
I understand. So, but,
So-
but they're also in the lower latitudes, like we're saying, if you're in the higher latitudes, you might have, especially if you had lakes and not a large extent ocean, if you had smaller lakes where you're concentrating that organic carbon, it might be easier to find.
Although he was saying it could be, I mean, I think that's kind of what they were saying. It was a kilometer down
Yeah,
from the lake. So that doesn't sound easier to me.
If it's deep
Well,
down.
those are probably
you're-
different things. I
you're
mean,
up-
one is it's surface lake when
Right.
they could live, and other if you don't have lakes, then actually in the pores like kilometer thick.
You mean deep aquifers, or what do you mean?
No, just I mean pretty much dry soil, but there is still some moisture in the pores.
Okay,
So you may have no like liquid water at all the surface, and still I guess you have life inside those. So.
Just to be clear, our resolution is 1 kilometer. Could be less. It's just that the first
Oh,
is a kilometer.
oh, that's the resolution
Yeah.
of your model anyway?
I mean, no, it's not 1 kilometer. It's yeah, okay. No, I was wrong. It's not.
Not one gram.
Oh,
Yeah,
okay.
it's. Oh, okay. Yeah, no. In
Hmm.
this scenario, it's much more optimistic than for some reason I thought.
Oh, it's less than.
It's nice.
The productivity is the up, it's very productive at the top.
Yeah, it's most productive at the top. This is where they get access to the H2 molecules,
Okay.
and the
makes sense.
lower strata they just have to live with the.
Yeah,
leftover.
I was wondering,
But yeah,
like, if
sorry.
you have such
It's okay.
deep pores, you have basically your hydrogen has to diffuse there, so you need some pretty high gradient to get it there. So
Why is it,
yeah,
why is it?
like
diffusing
on
here?
kilometer deep,
Yeah, it seems to be.
it would be difficult, I guess, to supply much hydrogen. But yeah, I see
Yeah,
that.
but depending on the scenario.
Orders of
But if
magnitude
you're
lower, so-
doing volcanic outgassing, wouldn't it diffuse up?
Yeah, so I guess it depends where you are, but yeah, the hypothesis there is that it's outgassed somewhere, mixes in the atmosphere and then diffuses.
Back
Yeah.
down.
Oh, and then
Yeah,
it goes back
Yeah,
down.
yeah, I guess it depends on your definition of outgassing. There is a, how they call, like, basically like outgassing in the sense of volcanoes, and there is also,
Yeah.
what's the term?
In-gassing.
In-
In-gassing.
In-gassing, yeah, I guess when it basically raised from the bottom. But you probably wouldn't get much hydrogen like with this in-gassing.
Well, wouldn't you, I mean, wouldn't you get kind of like, I mean, the whole crust is kind of like, it's not outgassing necessarily in a point source, right? It's all kind of diffuse, right? I mean, when
No, most likely it's probably a volcanoes, I mean, like how they call it, explosive volcanoes now. It's,
Well, not necessarily. If you've got flood basalts, it's going to be diffuse.
mm,
So it really depends on the eruption type.
And the Olympus Mons looks like a giant bunch of salt.
Yeah.
yeah, yeah.
Maybe the biggest one in the solar system.
Yeah.
Biggest single.
It's cool work, and I'm glad you came here because it gave me an opportunity to read this paper.
Okay, let's thank Antonin again. Thank you all online. Talk to you later.